Okay, so we are ready to start. Hi everyone. Welcome to Neuroelectrics webinar. My name is Rafał Nowak, and I will be your host today again. And today we'll be talking about bridging invasive and non-invasive neuromodulation in Parkinson's disease. And we'll discuss how DBS network mapping can be used to identify brain networks that may be targeted in a non-invasive way. And I'm very happy to welcome our guest speaker. Today we have with us Dr. Lukas Goede. Hi, Lukas. Good to have you here.
Thank you very much.
So just a little housekeeping before we get started. We'll begin with Lukas' presentation, and this will be followed by a Q&A session. So we'll have 45 minutes of the presentation and then 15 minutes for the Q&A. So please free to submit your questions at any time using the chat tool. So during the presentation, you can also start submitting the question that will be answered at the end of the presentation. And so yeah, let's go to the quick introduction. Let me quickly introduce our speaker. So Dr. Lukas Goede is a clinician scientist at Charité Universitätsmedizin Berlin. He began his career as a neurology resident in the movement disorders and neuromodulation unit at Charité in 2020. And from 2024 to 2025, he was a research fellow at the Center for Brain Circuit Therapeutics at Brigham and Women's Hospital in Boston, working with Drs. Andreas Horn and Michael Fox on DBS network mapping and multifocal TDCS. So his research focuses on identifying brain network through DBS network mapping and developing non-invasive strategies to modulate them. By combining clinical experience with neuron-based research, his final goal is to improve outcomes for patients with Parkinson's disease and other movement disorders. So welcome, Lukas, all yours.
Thank you very much. Thank you very much for the kind invitation and the kind words. I'm going to share my screen and hope that all of you can see my presentation correctly.
Yes, all correct.
Perfect. So today I'm going to talk about linking invasive and non-invasive brain stimulation in Parkinson's disease. As introduced already, I started working as a neurology resident and saw a lot of, specifically in the unit for movement disorders, and saw a lot of patients with Parkinson's disease before and after TDCS. And I think brain stimulation in general is a very good possibility for patients to improve their motor symptoms. And we were wondering if we can link this invasive standard therapy or established therapy with non-invasive brain stimulation to improve this technique further. So I'm going to talk about brain circuits linking invasive and non-invasive brain stimulation for a lot of different diseases, but we'll focus on Parkinson's disease. In the next part, I'll talk about deep brain stimulation network mapping as a method to identify brain networks that we can stimulate non-invasively. And then I'll talk about two recent studies that we published about network-targeted multifocal TDCS. At first, a randomized trial in patients with Parkinson's disease, and secondly, about a synergy between dopamine and brain stimulation, and how these different types of brain stimulation and levodopa may play a role together. So where does this idea come from that same circuits link invasive and non-invasive neuromodulation? Back from 2012, there was a study by Mike Fox who investigated outcome in patients with depression. And as we know, the subgenual cingulate is an area which has an altered activity in patients with depression. And we can measure the bold signal using functional MRI in this specific region and can compare it to all different areas across the brain and specifically the cortex. And it has been shown that the subgenual cingulate is anticorrelated to the dorsolateral prefrontal cortex, shown here as a black circle. And the colleagues Ann Weigand and Andy Horn investigated in a study led by Michael Fox how the outcome in patients with depression changed, and how this correlates with the connectivity to the subgenual cingulate. And they found out that the closer you get to the highly connected region, to the subgenual, or if it's more anticorrelated, the connectivity, there is a bigger clinical improvement in these patients, shown as the green circle here. And another example is a patient with another area that was stimulated using TMS in this study. And this patient showed worse improvement in depression. So this is a link about how interconnected regions, when they're stimulated on different sites, can improve symptoms a different way. And in this very comprehensive review, Michael Fox showed in 2014 in 14 different diseases how, so there was a review on different studies, and he showed how subcortical regions are connected to cortical regions that are also have been chosen as targets for TMS, for example, or also for TDCS studies. And in the rest of my talk, I'll mainly focus on Parkinson's disease, as we know that the subthalamic or also GPI targeted deep brain stimulation leads to symptom improvement in these patients, but also anodal TDCS, or excitatory TDCS over M1 leads to a benefit in patients. And I'll quickly summarize some studies, also back from 2006 in patients with Parkinson's disease trials, where an anode and a cathode, so bipolar TDCS, was used in Parkinson's disease. And for example, in this study, it has been shown that anodal TDCS over the motor cortex led to symptom improvement in patients with Parkinson's disease, measured in the standardized score. This is the UPDRS score. It's a Unified Parkinson's Disease Rating Scale. And with this score, we can measure different motor domains in these patients. So anodal TDCS versus sham led to symptom improvement. Cathodal TDCS didn't lead to significant symptom improvement, and also no significant changes when we compare it to sham stimulation. Another example was this first RCT by Benninger et al. In 2010, where it has been shown that multiple sessions of anodal TDCS over M1, so on 8 days within 2 weeks, led to a decrease in bradykinesia, one of the main symptoms in Parkinson's disease, and showed also a short benefit in walking time, but no significant effect on the UPDRS was shown in this study. And I summarized multiple studies. They had different outcomes. Some had a significant improvement, some only a significant improvement in gait, for example. Others showed no benefit, then only in bradykinesia, for example, but no effect in other motor domains. So a lot of studies have been done using bipolar TDCS in Parkinson's disease. Why we thought that it might be beneficial to not target only one point using TDCS, but rather a whole network, is this study from 2017, where the traditional approach of TDCS using one anode and one cathode, here shown in this figure, was compared to a network-targeted TDCS. So the network of the left motor cortex was targeted in the second part of the study. What the authors did is that they did a TMS pulse and measured the MEP amplitude on the contralateral hand. And after that, they applied TDCS with the traditional approach or the network approach. and measured the MEP amplitude using the same TMS pulse again, and could show that after network TDCS, there was a higher MEP amplitude compared to the traditional TDCS. And there are probably some plasticity effects in these patients, why the MEP amplitude was still higher about 1 hour after stimulation. So this study makes it promising to try a multifocal approach compared to a traditional approach in patients. And but how do we identify these optimal brain networks? So in this study, we have seen that the M1 motor network was used, so mainly the motor strip. But how do we identify other networks from other diseases? And to do this, we can use connectomic deep brain stimulation. So this is the idea comes from that point that we know that DBS is an established therapy and very effective therapy, a very effective brain stimulation method that leads to symptom improvement in patients, for example, with tremor or Parkinson's disease. And we can look at specific points where the electrode stimulates and use resting state functional MRI. So use the connectivity to different brain regions to identify these networks. And for that use, we don't need functional imaging of these patients. We rather use coordinates that we load into a human connectome. So a matrix of all interconnected brain regions that can lead to information that we can use. And we used a technique mainly introduced by Andy Horn, who supervised these studies that I'm going to present here. And for this purpose, we use LeadDBS software, an open source toolbox that can be used in MATLAB, and where we can localize the DBS electrodes. So the first step to really identify these networks is to learn from DBS which networks are stimulated and which lead to the best outcome in these patients. So briefly, what we do is that we have preoperative imaging and postoperative imaging, and we co-register these two images and normalize them in the next step to reconstruct the coordinates where the electrode is placed within the subthalamic nucleus in this example, or the GPI, for example, globus pallidus internus, depending on the cohort. And then we calculate or estimate the volume around the electrodes that is modulated. So depending on the current that is applied in these specific patients. And then we have the coordinates of the volume around the electrode, it's called VTA, volume of tissue activated, that is stimulated. And in the next step, we extract the bold signal within this functional connectome inside the VTA and correlate it to all other voxels in the brain. And what we get out of this analysis is a connectivity map, which is like a fingerprint of this specific stimulation. And this is shown here as well in our recent study where we used multiple different cohorts, where we, in the first step, again, performed the electrode localization, estimated the VTA, correlated these bold signals using the HCP connectome. This is an available connectome from the Human Connectome project. And then we have these different fingerprint maps from every stimulation setting. And we correlate this fingerprint in the next step with the improvement scores. So in a cohort with thalamic DBS, we had a tremor cohort. We used the tremor score. And in an STN cohort, so Parkinson's disease cohort with subthalamic nucleus stimulation, we correlated these fingerprint map values with the UPDRS score. And we receive a map that shows correlation values, and regions with a high value represent regions that are beneficial when they are stimulated using brain stimulation. And we showed that also across different cohorts, there might be an overlap for a specific symptom. So we think that there are symptom-specific beneficial maps, so optimal response networks in the brain that can be modulated. And so to summarize the first part, we have the deep brain stimulation where we know that it works really well in these patients. Or we also include, of course, patients where it doesn't work really well, and that is maybe connected to other brain regions. And we identified these optimal regions in the brain on a cortical level. And here we see that there's a cortical representation of points in the brain that are, or regions in the brain that are highly connected to the electrodes. And what can we do with these cortical representations? And we can, of course, try to modulate them from the outside. So we have to optimize a montage to stimulate the brain cortex from the outside using, for example, TDCS to finally modulate, or with the goal to finally modulate these regions. And we did this in this trial that we performed here in Berlin and published 2024. There was a pre-registered clinical trial. It had a double-blind crossover design. The examiners and the patients were blinded, and we performed blind video ratings of the UPDRS, so the standardized motor score, where we can analyze bradykinesia, tremor, and rigidity, but also gait. And we try to do our best to have it really as a blinded exam in these patients. And as a target, we used this RMAP, this optimal map, or we also call it Parkinson's disease response network. That was initially published by Andy Horn in 2017 in this paper. And we also reproduced this finding in 2021 across different cohorts. So we had an STN cohort, so a patient cohort with subthalamic DBS in Parkinson's disease, and also a cohort with pallidal DBS in Parkinson's disease. And they could cross-predict each other. So we were really sure that this network is a PD response network, irrespective of the DBS target that we use. So this part is all about identification of the optimal network, and that we tried to modulate using non-invasive methods, TDCS. And there was a study design. We did a power calculation, 0.8, and for that we included 23 patients, and we randomized these patients to one arm of patients who received sham stimulation on the first day and the active TDCS on the second day, or the other way around, active stimulation on the first day and sham stimulation on the second day, each for 20 minutes. And after that, we performed the UPDRS score, and also one hour later. But the main outcome was pre-stimulation scores versus post-stimulation scores. And between those 2 different days, we had a washout period of 24 hours to be sure that there is no longer lasting effect of TDCS, or that the effect would be so small that it wouldn't influence it on the next day. So in summary, these were our methods. We had our cohort of deep brain stimulation patients. We knew their outcome. We identified the connectivity profiles, correlated them with clinical scores to receive the map of the Parkinson's disease response network, so our target. And together with the modeling team from Neuroelectrics, we found an optimization to target these network the best way that we could. In total, we had 32 different possible channels, and the best montage was using 14 different channels to target this network. And then we applied multifocal TDCS with the star stem cap from Neuroelectrics. There were some safety rules in this cap, so maximum of 2 milliamps per electrode and not over 4 milliamps across all electrodes. And the best optimized montage was the use of the 14 active electrodes, which is shown here. Here are more details from our optimization. It's also published in this paper. So yeah, we would be happy if other groups reproduce our finding in PD patients. So it's shown here that we mainly focused also on the motor strip, but and also these other polarity of other electrodes around it, was the idea that that would really focus on our regions that we wanted to target in our network. And these were the results of the study. So the main outcome, MDS-UPDRS score after stimulation and after sham. So here in orange, that shows the improvement of the MDS-UPDRS in the whole group. And that was significant, and there was no significant change in the sham group. The effect was mild, so only mildly above the threshold of a clinical valuable improvement, but still an improvement after one session of TDCS. And here we can see the different subjects. So we also saw in some subjects an improvement also in the sham group, but also a lot of patients where no significant difference was shown, and in more patients it was shown that there was an improvement after the active stimulation baseline versus post-intervention. We compared this to the sham group, and for that we used the acti-sham protocol. This is a protocol where we also apply current all the time using the same electrodes, so that patients couldn't realize the difference between there was the idea behind it that patients don't realize a difference between active stimulation and the sham stimulation, because in some sham protocols you have just a ramping up and then no current and then just ramping down. And in this protocol, the idea is that we apply current all the time, but that the electric field doesn't target our network, and then the idea is that most current would shunt through the skin. And we asked also the patients if they could realize the difference between active stimulation and acti-sham stimulation. And 10 out of the 21 patients realized more tingling sensation on the stimulation day. Actually, 10 patients couldn't distinguish, and one patient realized more tingling on the sham day. So the current is a bit higher on the active stimulation day, but there was no significance in motor outcome when we compare those two groups. When we compare groups of subjects that they guessed correctly that was the stimulation condition, or based on the head sensation. So we think it's a real result and no placebo result in our study. So what we learned from this study, one takeaway is that we think that invasive stimulation informs non-invasive stimulation, but the question is, can we also turn this around and say, like, non-invasive stimulation can inform invasive stimulation? And luckily, some of our patients later received also deep brain stimulation. That was not part of the study. It was for clinical reasons. And we looked into the data. So again, from other patients, we had deep brain stimulation. We localized the electrodes, had the connectivity profile, calculated the optimal response network, and targeted the identified network using TDCS. And the question is, can we inform other DBS patients or later DBS in these patients, so in the same patients, based on the TDCS. In other words, does the TDCS outcome correlate with later DBS outcome in these patients? And these were the electrodes in these 10 out of our patients that received DBS later, so very well placed in the subthalamic nucleus. And we compared or correlated the relative TDCS improvement with the relative DBS improvement, and could see a significant correlation between the outcome of DBS and TDCS patients. And we explained two different cases within our study of patients who had a very good effect, specifically with tremor. So there was one example, and the other example was a patient with mainly gait problems who had no good effect in both stimulation protocols. So that led to the next analysis we did with patients. And we thought about, might the patient selection in deep brain stimulation be better when we have TDCS before DBS to estimate a later DBS outcome? And this is a very important question, because currently we can somehow estimate the later DBS effect. So we know, for example, for tremor, it's really good, or for specific motor parts, it's really good. But we currently think that when we have a very good effect using levodopa, we estimate a very good effect later in the DBS. But there are other factors which play a role, and there's not a real biomarker that we would have. So we analyzed the data based on this. And another important point was that subjects from our first study reported more improvement in the levodopa challenge after active TDCS. So they could measure it, but they told us when taking levodopa afterwards, they realized that it felt some things, like the muscles felt less stiff, or that they could move better or a bit faster. So we thought about and we had some data in most patients of a levodopa challenge, because we included these patients in our clinic, and usually in these visits we also perform a levodopa challenge, which was not part of the study, but we had the data for it, and so we analyzed it. And there was an analysis I did together with my colleagues, Patricia Zwarova and Bane Barnas, and led by Andy Horn. And we looked into a cohort of patients that received levodopa after both active stimulation and sham stimulation. So our study that I explained currently, and afterwards a levodopa test. And we had this levodopa challenge on both visits. So and we compared how well was the levodopa improvement on the sham day and on the stim day. And in the single data, we could already see that in a lot of patients, we found out that there was a bigger effect of the levodopa challenge on the stimulation day. So there might be an overlapping effect in these patients. And our theory is that all interventions like medication and also brain stimulation, invasive and non-invasive brain stimulation, in some way modulate the specific network in the brain. And maybe there are synergical effects in these different applications. So we analyzed this over the group. First of all, the baseline MDS, UPDRS scores, so the motor score, were similar on both days, the TDCS day and the sham days. No significant difference on these 2 days. And then we compared the scores after applying 200 milligrams of oral levodopa. And the scores decreased on both days, but more on active stim versus sham day. So the important thing is, in Parkinson's disease, so the idiopathic Parkinson's disease, there should be a levodopa effect in these patients, and this is why we included only patients with a clear diagnosis of Parkinson's disease and no atypical Parkinson's disease. So there was an effect on both days. This is what we expected. But then we could see that on the day where we had active TDCS, the improvement scores were higher compared to the improvement of levodopa on the sham days. And when we also in the next step compared these improvement scores with the later DBS improvement scores, because as I told earlier, we know that DBS improvement in some studies correlates with levodopa improvement. We looked into it again and in a subgroup of patients here and could see that there was an effect or a correlation which missed the significance value of p or the p value, and but there was a positive but not significant correlation. Same for DBS improvement and TDCS improvement in this subgroup of patients. But when we summarize these two values, so the levodopa improvement and the TDCS improvement in a linear model together, it showed in these 10 patients a positive estimation that was significant in these patients. Maybe importantly, it's very important to say that there was only a small cohort of patients, and it was not the primary outcome of the initial study. So also the comparison of DBS effects and TDCS effects, the study was not powered for us. It was an explanatory analysis, but we thought it's really important to talk about these results because it is a very unique cohort of patients who received TDCS, the levodopa challenge, and received DBS. So I think it's a very good point to discuss and to start working on. We have to discuss that some patients didn't show any effect after TDCS, but I mean, this can always happen, but why did some improve also after sham stimulation? And I think one important point is that in Parkinson's disease, there are different days, and we have some fluctuations over the day and also on different days. So maybe one important side note is that in the first study, patients were really in the off state. So they didn't took any medication. So we don't think that there is an influence of medication. So they paused it for overnight. But in general, we need more studies to reproduce it and also see how much effect is there after sham stimulation versus stimulation. And we had just one session of TDCS. We need to discuss, are more sessions needed for longer effects, for example, and what's the best amplitude to stimulate? We choose the maximum possible amplitude that's feasible in these patients and that's safe, of course. And so 2 milliamps per electrode is like a very, very often used amplitude in patients. And we just are not sure about what's the best amplitude and also how many sessions do we need and how long should the stimulation be. There's work which showed that we should apply TDCS for at least 13 minutes, for example, and also on multiple sessions. And but this is I think part of the future research, and I'll show one study where we're trying to do this. And we also identified further networks. For example, so we talked about Parkinson's disease. We have a tremor response network and also in OCD, so obsessive compulsive disorder, a network that can be targeted using a non-invasive brain stimulation. And more networks, functional networks are published. So we learn more and more about brain networks, and maybe we can target them non-invasively. So we can translate a lot of different networks to TDCS, and that needs to be tested. Another further application is a study led by Michael Fox at the Brigham and Women's Hospital, I'm involved in. So I initiated it together with him in Boston. This year, it's a safety and feasibility trial of a tele-supervised home-based TDCS in Parkinson's disease. So we worked together with the modeling team of Neuroelectrics again to be able to target this network the best way with less electrodes that are available in the at-home setting, because using a lot of gel electrodes in clinics needs some experience and needs someone else who really learns how to keep the impedance values low to really be able to have a good setup in these patients. And so we thought about how to make it as simple as possible, but as effective as possible for the patients. So we finally, or Neuroelectrics came up with this optimization after working on it. And we started this study, and it's still ongoing. So we need 2 more subjects in this study, which is here pre-registered at clinicaltrials.gov. And yeah, hopefully we can complete it soon. And what we are doing in this study is that we have 7 sessions within 1 week at home. So we telesupervise these patients. We do motor scores pre and post stimulation, so before this week and after this week. And we have a lot of questionnaires about the feasibility of this trial, because I think one of the pros for non-invasive brain stimulation is that we can really apply it at home without any surgery. And yeah, we hope that might lead to a bigger effect in these patients. So with that, I'm summarizing my talk and what we did over the last years. We use DBS to define optimal brain networks, and then we tried to use multifocal non-invasive brain stimulation, and this led to motor improvement in Parkinson's disease compared to sham. The TDCS outcome was associated with DBS outcome in this subgroup of patients, and TDCS together with the levodopa challenge may estimate the later DBS outcome as a small exploratory analysis. And yeah, we are looking forward to look more into it in future research. And with that, I'm in the end of my talk. Thank you very much, everyone, for your attention. And I want to thank all my colleagues, both at Charité, Universitätsmedizin Berlin in the movement disorders group, and also at the Center for Brain Therapeutics in Boston, where I spent 1 research year, and my mentors, Andy Horn, with whom I did most of the work, and also for the support from Andrea Kühn and Michael Fox, who led the centers where I worked in and gave a lot of input to the studies. So thank you very much.
Thank you very much, Lukas. Thank you for this great presentation, very promising preliminary results. We are ready to take some questions. Actually, we have already some questions. So we have some question from Christoph regarding, I think, the last part of the presentation, mainly regarding, we were talking about network maps like RMAP, if there is a non-invasive way to create the network map for other conditions or general mental state, similar to connectivity analysis, what you were mentioning. So you're mentioning like Parkinson, in Parkinson, in essential tremor, in OCD, and at the general population level. So like the first question would be if we could personalize this, and the second question, if there is also an option to create these maps for other conditions or general mental states.
Yes, so yeah, this is a great question. And the first point is that I mainly worked with DBS network mapping, so using information that we receive from implanted DBS electrodes and the outcome in these patients, so in the specific cohort. And for that, of course, we don't have, or we have a lot of there is a lot of data exists in different neuropsychiatric disorders, which can still be analyzed to identify more networks. But on the other hand, we can also learn from other lesions, and this is, I think, mainly part of the of Mike Fox lab, who uses lesion network mapping. So what we learn from lesions, so not implanted electrodes, but lesions. So for example, stroke lesions, what happens, how does symptoms change? So we can also, or they have a lot of work on identifying brain networks that also led to symptom improvement or that are associated with specific neuropsychiatric symptoms. And the second part about the personalization is that, and I think for that, a very, very nice example is the first trial that I presented in where we know that a specific region in a patient can be, or let's say in a specific part of the brain in a patient with a disease, we see a difference in bold signal, for example, the subgenual cingulate in depression, patients with depression. And we can look at the regions that are highly connected to it and can use also individual MRI to really map this region and to target specifically this region using, for example, TMS. And this is exactly what they did in this analysis to see how close we go to an optimal point in these patients and how this correlates with clinical improvement.
Okay, thank you very much. Actually, this covers already the second question regarding the individualization and personalization. And we have another question. Yeah.
Maybe one more point.
Yeah.
So we used the EEG nomenclature using an EEG cap to navigate our stimulation. But of course, this is one more limitation that you could take an individual MRI scan because brains are different and then target specific points and using a navigation system to really be sure to target in the correct way. But it's always, in between. I mean, how focal is this type of stimulation when you using non-invasive stimulation? And we thought the EEG coordinates using in patients is the practical way because also patients were in the off state and it already took a long time to do the montage of the cap and then do all the tests and let them stay 1 more hour in the off state. That was already really hard, but I think you can also individualize it using individual MRI scans.
Perfect. Thank you very much. And actually, this is related to the second part in the second study. What preparation of levodopa was used? And we know pure levodopa has low bioavailability.
Yeah, so we used levodopa benzerazide as solution. So it's an oral medication that you can put into water that is soluble, and then it's levodopa benzerazide. This is how we perform the standardized levodopa challenge here in our center.
And the follow-up question, if there were any idiosyncratic reaction in the range of psychosis shown in any participant?
Yeah, it's a great question because I think we could also maybe induce or make some psychiatric symptoms worse. We haven't seen it in our cohort. It was more a positive effect in most patients that they felt better afterwards. Not sure if this is a placebo effect or that was not part of our outcome, but it's always important to mention and to be sure that you don't target a network that can lead to worsening of any symptoms. Of course, it's very important.
And the third questions regarding still levodopa is a chance directly instilling a levodopa with DBS in the brain.
Stealing?
Yes.
No, I think the mechanism is different. So we just think that it influences similar networks in some way and leads to a change in the electrophysiological markers. But yeah, I think it's rather that it complements each other to improve or that there is a synergy between those two techniques, or the medication and the stimulation.
Okay, so like from the general point of view, could we like hypothesize that the network related to DBS, network related to TDCS, and then dopamine network somehow overlap based on these three different approaches?
Yes, this is what we hypothesized, but yeah, this is one idea why it could lead to bigger effect, and also in this model together. But yeah, as I said, it's still some exploratory results in there, and yeah, but it was the idea after what patients told us after finishing our study, why we came to this idea to test this.
Okay, another question, if you know about other group investigating the use of TDCS or TACS to predict the efficacy of DBS treatment. Other group than yours, and the group in Charité and the group in Boston.
No, I mean there is this overview that I showed, and I summarized it also in the movement disorders paper in the supplementary material about TDCS studies in Parkinson's disease. But I don't know about a study using this DBS informed maps so far. But yeah, also this montage and how we or where we stimulated with the values of the current that we used is also published. So so far I haven't seen it that someone else did this, but I hope that other people try it.
And there is room for collaboration, I guess.
Of
And
course.
actually, the data are in the publications.
Yes.
The movement disorder publication. There is a montage. I'm not sure if there are current videos of the supplement. You can find the videos for every position, I guess. So you can reproduce perfectly the
Yes,
montage for the
exactly.
TDCS.
And always reach out for collaboration, of course.
Okay. A question from Edward Cortin. Hello, great talk. Thank you very much. I have looked at the multifocal TDCS effect on beta power in patients that have sensing-enabled DBS. Actually, this is the question, sorry. If you have looked at the multifocal TDCS effect on beta power in patients that have sensing-enabled DBS, I
Yeah.
guess it's related to EEG beta power.
No, it can. So it's actually a very great idea, and I would love to do it, but we haven't applied TDCS, and so it's probably about patients who have already implanted DBS with sensing possibility. So we just applied TDCS on patients without deep brain stimulation. I would love to look into data like this, how we really change the beta activity in these patients. But I think it's for safety reasons probably not possible to stimulate patients with DBS using TDCS. But it would be great to have also these kind of data.
Okay, thank you. Next question from Dart. After prolonged levodopa treatment, 5 years or more, dyskinesias appear. Do you foresee TDCS as a means to decrease, control this outcome and improve clinical results?
Yeah, so it's also a great question because we know that deep brain stimulation is a good treatment option for patients with Parkinson's disease with dyskinesia after a couple of years. And so one idea how we can test this would, I think, the at-home stimulation, non-invasive stimulation for TDCS. So I think after we haven't looked into dyskinesia after this one stimulation, but I think we need more data from patients who receive TDCS at home to see if there is an improvement. So I cannot answer it right now, but I think that would be after So if it's safe and feasible to do this at home TDCS, that would be really interesting to look into.
Okay, thank you very much. The next question is regarding from Paco regarding specific stimulation structure. How does from your experience, how does the temporal structure of stimulation, intensive short-term protocols versus spaced long-term sessions, affect outcomes and neuroplasticity in Parkinson's or similar disorders? Do you see evidence favoring one pattern over the other for durable effects?
So I know that other groups did a lot of research on how long or how does the MEP amplitude changes after specific durations of TDCS, and I think it changes for a longer time for after applying TDCS for at least 13 minutes. There was a cutoff I read about, but to be honest, I'm not sure, and I think there is no data about what is the optimal length of one TDCS session. So most studies use 20 minutes. Also, we use 20 minutes. And also another question is how often should we apply TDCS and when? So maybe applying TDCS and before a washout, applying it again. So that is one of the ideas, but I think it's not really known as far as I know.
Yeah, but yeah, for sure, we know that the 20 minutes in one session is very, very short-lasting effect. So if you will do repeat this over many sessions, that did what probably you are doing in this home study, the longer-lasting effect would be more than expected. Okay. Actually, we have friends, your friends from Germany. The group of Bettina Schwab in Hamburg University Hospital is working on doing DBS while applying TACS as a closed loop. So we have, so there is another group, and actually doing closed loop stimulation based on DBS and alternating current stimulation.
Okay. Yeah, thank you very much. Really, really interesting. Yeah, we only used TDCS currently, and but this is exactly what we need, like this closed loop between non-invasive and invasive stimulation, not only in the analysis that we did, so one after another, but at the same time, really looking forward to see the results.
Perfect. Thank you very much. Just to summarize, we have very good results regarding the network definition. We have some signals of possibility of using TDCS as a, if I can say this, predictor biomarker of the selection for DBS patients, the responders versus non-responders. And yeah, can we say this?
Yes. Yeah, we have some hints for it. Yeah.
Okay. And there is somehow correlation between the TDCS and response to DBS.
Yes, and maybe even better when we also combine it with for example, levodopa. So when we have so the goal could be like to really build a model after which we can estimate the DBS outcome in a better way. And maybe TDCS is one part of it.
Okay, and finally, maybe going even more to personalization and profiling patients based on some phenotyping, who will be the most likely to respond to network targeted TDCS? Actually, you have had also variability. Yeah, I know the N is quite small, but there was also different variability based on the different symptoms.
Yes.
Okay, so yeah, go ahead. Sorry.
Yeah, so from our study, what I realized, but we didn't have enough patients to look into these different subgroups. But that one point was that patients who had a mild Parkinson's disease or were at the beginning of the disease responded better to the treatment. So for people who work with Parkinson's disease know that after couple of years having this disease and being in the off state can cause really severe motor problems. And I think that TDCS wouldn't be effective enough to really lead or bring people out of this off state. So it can show an improvement, but I think it works better for patients with milder Parkinson's disease, or are at the beginning to really see an improvement that is significant also for the patients. So this could be a subgroup where we could focus on, I think, for the future. But this is just a gut feeling or some experience we had within the study. I don't have any, and I don't have enough data to really show it.
Perfect. Perfect. Thank you. Thank you very much. Thank you for great presentation, and congratulations on the preliminary results, and good luck with the next steps. Thanks everyone for joining us today, and I hope you'll join us again for the next webinars. So you can find always the information on our webpage, neuroscience.com, and see you again soon. Have a great day and take care again. Goodbye. Thank you, Lucas.
Thank you very much. Bye bye.